it's good to talk about photosynthesis. There's not that much photosynthesis going on at these cold temperatures, by the way. So we've seen this overview of what happens in light reactions. We're now going to look at some details. Okay, so leaves are green. Why are leaves green? Leaves are green because if light hits the green leaf, wavelengths other than green, so red, blue, yellow, are absorbed, but green light is transmitted through the leaf or is reflected back. The leaf is not absorbing so much in the green area. And this is due to pigments, and the main pigment is chlorophyll. Chloros is Greek for green. Chloros is Greek for green. So chlorophyll means the green pigment of leaves. Phyll is leaf in Greek. So chlorophyll is very important in photosynthesis, and how can we decide that that's the case? If we take a spectrophotometer, which is a machine in which we can put a sample, we can shine light on it at various wavelengths, and we can measure how much of that light is absorbed. We can put different isolated molecules in there, and as a function of the wavelength that we're giving, we can measure how much of the light energy is absorbed. And we're seeing that chlorophyll, of which there's two major kinds, chlorophyll A and B, is absorbing here a lot in this blue region and in the red region, but not a lot in the green region. We're seeing that there's another pigment that's abundant in leaves, but not as abundant as chlorophyll, and that's carotene. And carotene is the stuff that makes carrots look orange. The carotene absorbs more in the green and blue regions of the spectrum of visible light. So if we now do another experiment, and we're putting a leaf in a device where we can measure the amount of CO2 that's being fixed. And we're putting light on the leaf of different wavelengths, and then we're looking at the highest activities, or we're looking at the activity as a function of that wavelength. So we're seeing that in the blue to early green, bluish wavelengths, so right here, we're seeing high activity. That's the action spectrum, is our red line. And in the red wavelengths, we see high activity. So we see high activity in the red and the blue region, not so much in the green region. That's the activity of the intact leaf for fixing CO2. So this action spectrum of the leaf for total photosynthesis matches mostly with the absorbance spectrum for chlorophyll A and B combined. And therefore we can say that chlorophyll seems to be the pigment that is mediating light absorption for use in photosynthesis. So I like to say, based on this experiment, chlorophyll is guilty by association. Okay. Well, we know that plants without chlorophyll don't photosynthesize. There's more direct evidence for the involvement of chlorophyll in photosynthesis. This is the structure of chlorophyll. It has this long fatty-like tail, an isoprene tail, which just means that this is a molecule that is heavy in the membrane. Then it has this ring structure that's called a porphyrin ring, within the center of magnesium ion. This structure, porphyrin, with an ion in the center, is something we've seen before. In the case of heme, which is present in cytochromes and in hemoglobin, we have iron in the center, but here we have magnesium in the center. And this set of alternating double bonds, single bonds, that's a system of conjugated double bonds there, which allows this molecule to absorb light over a broad set of wavelengths. The conjugated double bond system gives that property. And when light is absorbed, what we imagine goes on is electrons that are in the orbitals of this conjugated ring system take up a position corresponding to a higher energy. You can think of that as an electron moving further away from a nucleus. But in chemical physical terms, we have to say the electrons take up an orbital that corresponds to a higher energy level. So when a photon of light hits this chlorophyll molecule, an electron can become excited. Now, chlorophylls absorb the light, but the real action of photosynthesis involves chlorophylls that are associated with proteins at specific sites called the reaction centers. And we know that there's much more chlorophyll in the leaf than what is present in the reaction centers. The majority of chlorophyll is not in the reaction centers, it's somewhere else. What is it doing? It is sitting associated with other proteins that we call the light harvesting complex proteins, indicated as these green circles here. In this lower diagram, imagine we're looking down on the thylakoid membrane on the surface. We're seeing there a reaction center in the middle, which is surrounded by other proteins that we call the light harvesting complex proteins. And these proteins have a lot of chlorophyll bound. The light is absorbed by these reaction center chlorophylls and is then transmitted from chlorophyll to chlorophyll. And energy is transmitted from chlorophyll to chlorophyll until it ends up in the reaction center. That process is called resonance energy transfer. It's not really one chlorophyll getting excited, emitting light, and another chlorophyll absorbs it. It's direct transfer of that energy from one to the other chlorophyll. And that makes that overall each reaction center has access to a large surface area for absorbing light energy, photons. Now there are two reaction centers, which we call photosystem 2 and photosystem 1. So, the job of chlorophyll is to absorb light energy. Now let's remember, the energy in light comes in packages that is of an appropriate size to do chemical reactions without being destructive. If we go to lower wavelengths, higher frequency, go to the UV area, you get into radiation that would have destructive interactions with organic molecules. If we go beyond the red, then we get into the range where the energy in a photon is too low to do anything. So the reason that we see light and that plants do photosynthesis using visible light is related, I think. So what happens in the photosystem? There are two photosystems. They're called photosystem 1 and 2. 1 was discovered first, but 2 is the one that first acts in photosynthesis. So we're considering photosystem 2 first. Photosystem 2 sits in the thylakoid membrane, and it has a lot of these light harvesting complex proteins associated with it. This is where a lot of the chlorophyll is bound. There are two important chlorophylls in the reaction center that we call the active site chlorophylls. They're also known as the P680 pigment, absorbing maximally at 680 nanometers. So this is really the main business part of our reaction center. The energy is transferred from the light harvesting complex antenna chlorophylls to this reaction center chlorophyll. And what then happens is the reaction center chlorophyll gets excited. An electron is in an orbital with higher energy, and from there it can leave the chlorophyll and it can be transmitted in a small electron transport chain. First, it is transferred to a chlorophyll-like molecule that we call phytophiotin. This is the one, phyophytin, sorry. Phio in short. And then it's absorbed, the electron is then accepted by a quinone. And this quinone is called plastoquinone. It's very similar in structure to the ubiquinone that we've seen in the mitochondrion. This quinone is fixed in the reaction center, and it donates ultimately the electrons to another quinone that is mobile and that can diffuse away. So what happens? Light is absorbed, an electron is excited, that electron can then be transmitted and be lost from this chlorophyll in the reaction center, transmitted via short electron transport chain, leading to the reduction of a quinone, plastoquinone. It takes 2 electrons to reduce that quinone completely. So now we have a problem because the chlorophyll in the reaction center has lost an electron, it's therefore positively charged. That's unstable, it needs to get an electron back, and it gets that electron back from water. Not directly, but via an enzyme that we call the water splitting enzyme. Now there's a little bit of a problem here, and that we need 2 water to be split in order to form a full oxygen. And that reaction involves the liberation of 4 protons and 4 electrons. But light can only kick out 1 electron from the chlorophyll reaction center at a time. So we need a way to store these electrons from water, and that's the big achievement of this water splitting enzyme. It uses manganese ions to temporarily store those charges. And so we can see that for the formation of oxygen, we need actually 4 sequential electron transport events, which we can see if we give short flashes of light that allow this reaction center to only once boost the energy of an electron. So we start at this point here, and giving 1, 2, 3, 4 bursts of light, flashes of light very briefly, and only at the 4th flash do we see that oxygen is released. So what that tells us is that that oxygen is formed using 4 electron transport events here in this reaction center. So, photosystem 2 has a water splitting enzyme. It has chlorophylls in a reaction center. The chlorophylls absorb light energy that boosts up the energy level of an electron, which then can be used to reduce another component, ultimately reducing a quinone. The chlorophyll becomes positively charged in that process. That electron is replenished ultimately from water. This is something very complicated that nature achieved at some point during the evolution of life on the planet. So what happens again with the chlorophyll? We have the chlorophyll that we call the reaction center chlorophyll, P680. Light energy allows an electron to become excited. That's indicated by this star. This is now a high energy electron which can tumble down an electron transport chain, and via FiO it then ultimately reduces a quinone. That creates a positively charged chlorophyll, which has to get an electron back, and it takes that electron from water. The chlorophyll goes down to the ground state. It cycles like this. There's evidence for all of these steps because we can actually observe using EPR and other methods of physics. We can observe these excited states and these charged states of the chlorophylls. But it takes a thing that can measure in very brief periods of time. All right. So the job of photosystem 2 is really to reduce plastoquinone using electrons taken from water. Those electrons in water have low energy in association with oxygen. Those electrons by a special water-splitting enzyme and the chlorophylls are stolen from that oxygen in water, given to plastoquinone. This is how the quinone gets reduced. These two oxygens take up an electron and a proton, each has a full hydrogen. So that's the full reduction of plastoquinone. This is really the same as what is going on with ubiquinone in the mitochondrion, which is reduced by complex 1 or complex 2, remember? Okey-doke. Now, what's special about photosystem 2 is that it can do this charge separation and steal this electron from oxygen, and at the same time it can reduce a quinone, which requires a high amount of energy, so a low redox potential. Here, this high redox potential means a very electronegative group, a group that wants to steal an electron so much it can even compete with oxygen. The combination of both the very high redox potential and the low redox potential in one protein, which has the two sites or the two vastly different redox potentials close together, is something that's very difficult to achieve. Because you can imagine an electron right here really wants to fall back down to water, but that's prevented here. The system is driven by light. That's a huge accomplishment of life to do that. Alright. So I call this the big trick of photosynthesis. This is something that happened only once during the evolution of life on the planet. There's only one photosystem 2 water splitting enzyme. There's not another solution that we know of for the problem. But as soon as nature had invented this photosystem and could do this, it was such an advantage that it was used a lot. And for 2 billions of years, this process dominated life on the planet. Okay, so the quinone is reduced. What happens with it? The plastoquinone formed as reduced in photosystem 2 can go in an electron transport chain. And what it does is it reduces a complex that we call the cytochrome BF complex in plants. And this is very similar in succulent composition, in architecture, and is identical in function to the cytochrome BC1 complex that is active in the respiratory chain of mitochondria. This is essentially complex 3 of the mitochondrion. It's functionally the same thing and architecturally almost the same thing. They're the same cofactors. So the way this works is as the quinone donates its electrons to this complex, protons are moved across the membrane. Because the protons were taken from the strobal site when plastoquinone was reduced, and they're donated here to the phytoid lumen site. So this is redox loops here that make the protons go across. Now we need to get rid of the electrons, and there's a molecule called plastocyanin, this small copper-containing protein that's blue, that takes the electrons from this B6F complex. Plastocyanin is therefore completely equivalent in function to cytochrome C in the respiratory chain. In fact, there are bacteria, cyanobacteria, that photosynthesize that use cytochrome C in this position. They don't use plastocyanin. Honestly, plastocyanin was invented by life, or taken to be used in this function by life 2 billion years after photosynthesis was invented. So cytochrome C was used there first. It was later replaced by plastocyanin. Alright. So plastocyanin donates electrons to photosystem 1. What is photosystem 1 doing? So remember, this is an electron transport chain. It's proton moving, proton pumping by redox loops. So we need photosystem 1 because right here we're generating a proton-motive force that can later be used for the synthesis of ATP. But we also need to reduce in power. So how do we get the NADPH to be formed? The reduction of NADP+. That's the job of photosystem 1. It's the first photosystem that was discovered. That's the second photosystem that acts in the electron transport chain. Photosystem 1, similar to photosystem 2, has active site chlorophylls that pair there. They have a maximum absorbance at 700 nanometers. That's why we call it P700. If light energy hits this chlorophyll pair, or an electron is excited, that electron can go in an electron transport chain, and ultimately that leads to the reduction of NADP to NADPH. So 2 electrons are needed for that. The chlorophyll that has lost its electron here in the active site, after that electron was excited and loses that electron, that chlorophyll gets its electron back from plasticine. So, very similar to photosystem 2, there's a cycle where chlorophyll is excited, then loses that high energy electron, and then it gets the electron back. Photosystem 2 gets the electron back from water, photosystem 1 from a protein, plasticine. Okay, the job of photosystem 1 is to reduce NADP+. How are we doing? Good. Right. Now, the strongest oxidant known in nature is that water-splitting enzyme of photosystem 2. But right here, at the reducing site of photosystem 1, and really we can say that in the photosystem, but at the surface here, we still have a strong reductant. That is the strongest reductant that we know in nature. It's a very strong reductant. Okay. So what goes on with electron transport can be plotted in a different way. We can look at electron transport from a perspective of energy or the perspective of standard redox potentials. So we start here with water. Water has a low energy in its electrons. If we have a very electronegative group in a reaction center with a positively charged chlorophyll, we can take an electron from water and we get a neutral ground state chlorophyll. Light energy can boost up the energy level in an electron, in a chlorophyll. That chlorophyll can then lose that high energy electron to an electron transport chain. We tumble down, losing energy all the time, as protons are pumped by the mechanism of plastoquinone and the cytochrome B6F complex. Our electrons arrive at photosystem 1, but they haven't lost all their potential energy. So if a little bit more potential energy is added, we're ending up with a very high energy electron, which is a very strong reductant for that reaction center, so that can reduce things that are hard to reduce, such as NADPH. Because NADPH wants to really lose its electrons quite strongly. It's a strong reductant, but this is an even stronger reductant. So by having the 2 systems in series, we can overcome the energy difference between water and NADP, which is a big difference, and at the same time lose still some energy along the way in order to drive the synthesis of ATP. So the small trick of photosynthesis is to place 2 reaction centers in a series, which allows life to overcome that difficult electron, difficult potential energy difference. So on this scale, what we're looking at here, the delta G, has a high energy here on this scale on the top, and on the other end we can say we have the standard redox potential. So my arrow was lost. So at the bottom here, that's the highest standard redox potential. Because electrons flow to ever-increasing redox potential, remember that? So a photon has something like 42 kilocalories per mole. That's the photons that are absorbed by the chlorophylls in the reaction centers. To form NADPH, that's roughly 53 kilocalories per mole. 42 is not enough. We need 2 reaction centers. You can boost the energy level of the electron twice. And you have enough energy to reduce NADPH. Are we good? Alright. There's a figure in the introductory biology book by Campbell that I like, and that I've pulled up here. It gives an analogy for this process, and it's the hard hat workers analogy. Now I like the detail. You see the guys that do the hard work here? They have no sleeves on their shirt. They're doing the hard work, the sweaty guys. And the guys that are capturing the electrons here, that are doing the light work, they have a little bit of a more classic t-shirt. See that? So imagine there's light energy from a photon is used to kick up the energy in an electron. So it can be captured, and with a high potential energy, it can then be tumbled down an electron transport chain for the generation of ATP. Then it ends up at a second place, but not at the bottom. And at that second spot, its energy is further boosted by the light photon, particle of light. And then that electron is captured at the reducing side of photosystem 2. And it's allowed to reduce NADP plus to NADPH. Okay, so on this scale, ever increasing energy when we go up, and when we go down, we have ever increasing redox potential, more electronegative groups towards the bottom. So right here, photosystem 2 has a standard redox potential that's high enough to split water. And right here we have a standard redox potential that's low enough to allow us to reduce NADPH and subsequently carbon dioxide. I like that image. So, 2 photosystems in a series make it possible to synthesize ATP because the proton gradient that's produced here, those protons flow via the ATP synthase to give the generation of ATP from ADP and PI. The ATP synthesis occurs in the stroma, the equivalent of the cytoplasm for the chloroplast. It is the cytoplasm of a photosynthetic bacterium. At the other end, photosystem 1 drives the formation of reduced NADP. Alright. Now sometimes we need more ATP and less NADPH. So what can we do? Well, it turns out that photosystem 1 can not reduce NADP, but it can also choose to reduce the BF complex again, essentially by giving electrons back to the plastic quinone pool. And this involves another complex that's not in here, which is really a complex that functions like complex 1 in the mitochondrion. So that complex 1-like component in the mitochondrion can then lead to the reduction of the quinone pool, which with cytochrome B6F, that's a complex 3 in the mitochondrion, can generate more of a proton gradient, which is used to make more ATP. So in this scheme, the electrons are flowing around via the BF complex to plasticine into photosystem 1 and back. We call this cyclic electron flow. There's a lot of debate about how much cyclic electron flow there is relative to linear electron flow. Linear is from water to NADPH, using the 2 photosystems. And the estimates vary that this is between something like 5 to 20% of the total electron flow. But the important point is it helps to relatively make more ATP over NADPH. So this is a mechanism to adjust the ATP to NADPH ratio, cyclic electron flow. Now there are some bacteria that do not photosynthesize, but they have photosystems. They have a photosystem that functions like photosystem 1, and they have a B6F complex, so something like complex 3. And what these bacteria do, together with the cytochrome here, instead of plastocyanin, they have this cyclic electron transport, and they're using light energy to generate their ATP. There's some bacteria that do this. The purple sulfur bacteria, for instance, do this. Okay. So, to summarize, how do we make ATP? Electrons from photosystem 2, the primary acceptor there, we get a reduction of a quinone at a site that we call the QB site. Plastoquinone is reduced. These electrons go in an electron transport chain. Energy is released at the BF complex. So that energy is used to pump protons over the 5-part membrane. The direction of proton pumping is from the stroma into the 5-part lumen. The protons can diffuse back via the ATP synthase, and in that way ATP is formed. This is ATP that is formed under the influence of light, and ATP, that means ADP accepts phosphor, that's a phosphorylation, so we call this photophosphorylation. To distinguish from oxidative phosphorylation in the mitochondrion, and we also have substrate level phosphorylation, remember, in glycolysis. So we have 3 types of ATP formation: substrate level phosphorylation, oxidative phosphorylation, and we have photophosphorylation.